Okay, in this lecture, I'd like to think about the stages of X chromosome inactivation. So we know that X inactivation doesn't happen instantly, but instead happens progressively over a period of developmental time. And during this time, we know that there are specific stages that X inactivation needs to go through. So the first stage of X inactivation is counting. So what I mentioned earlier was that in karyotypically abnormal cells, we know that there's only going to be one X chromosome that remains active per diploid set of autosomes. And so the cell must have some mechanism to count how many X chromosomes there are in relation to how many autosomes there are within the cell. And this is the mechanism of counting. The second stage of X inactivation is choice. So I mentioned imprinted X inactivation in the last lecture. And clearly here, there is no choice. The choice has already been predetermined that the paternal X chromosome will be silenced. However, in this instance, choice means that in random X inactivation, there must be a choice about which of the two X chromosomes in a normal female cell will be the one that's selected to be inactivated. The third stage of X inactivation is the initiation. And really this is all about the expression of that long non-coding RNA that I mentioned in previous lectures called Xist. Xist expression comes from its particular location on the X chromosome known as the X inactivation center, which we'll talk about in future lectures. After this initiation of Xist expression, then the Xist needs to spread to cover the entire X chromosome that will become inactive. And following on from that, we need to have establishment of the silencing marks. And finally, after that, we will have the maintenance of these inactivation. And maintenance is, of course, perhaps the longest lived phase of X inactivation, because while the first few stages happen just within a few days in the embryo, maintenance must occur for the life of that female organism, and so can be occurring for many decades, up to perhaps 100 years. So while we'll go through many of the molecular events that are involved in each of these stages, I think the really key thing to understand and remember is that the way X inactivation happens is that there's a progressive layering of an accumulation of epigenetic marks. And it's by this progressive layering of redundant epigenetic marks that you can ensure this mitotic heritability and the incredible stability of X inactivation, and that's why it can last for the lifetime of an organism. So before going into all of the details for each of these different stages of X inactivation, the most important thing to remember is that it's really all about Xist. So Xist is the critical determinant for X inactivation to occur. This long non-coding RNA is essential for X inactivation. And so the first few stages of X inactivation, counting and choice, are really about getting Xist expressed in the first place. And the latter stages of X inactivation are turning this expression of Xist into transcriptional silence. And so just keep this in mind at all stages. So before we go any further, let's have a little refresher about what Xist is. So as I said, Xist is the critical determinant for X inactivation. Without Xist, nothing else can happen. So it's a 17 kilobase long non-coding RNA. It's spliced, just like a normal RNA would be. It's polyadenylated, but it's constrained to the nucleus. It isn't exported from the nucleus to be made into protein, and indeed there's no open reading frame to be found, so there's no protein product that's produced. We know that Xist is expressed from just one of the two X chromosomes in a normal female cell, and it coats that X chromosome in cis. That is, it coats the X chromosome from which it is expressed, but doesn't act on the other X chromosome. So the first detectable event in X inactivation is indeed the expression of this long non-coding RNA. So here, shown on the right, is an image that I've shown you before, showing the DNA, first of all, in the nucleus, which is stained in blue. And then we also have, shown in yellow, the Xist DNA itself. Of course, there are two pieces of Xist DNA, one on each of the two X chromosomes. So in addition to these two Xist DNA foci, which you can see with the very small yellow dots that are shown indicated with each of these arrows, you can also see the Xist RNA, which is stained in red using in situ hybridization. But this Xist RNA is not found on both X chromosomes, but instead just found on the inactive X chromosome, where it forms a cloud. So it's not a punctate RNA like you would expect if you just had a small amount of RNA being expressed and then being exported, but rather it's being expressed and retained in the region from which it's expressed. And so it forms this cloud which actually coats the entire X chromosome, which will become inactive. So let's now think about X chromosome counting, this first stage of X inactivation. Well, there still isn't a lot known about X chromosome counting, and over the years there have been many theories about how this might occur, many of which have been controversial. But I think now the consensus is that there are both X-linked factors and autosomal factors, and it's the balance between these X-linked factors and the autosomal factors which allows the cell to be able to count how many X chromosomes there are compared with how many autosomes. So far we know essentially nothing about these autosomal factors, but we do know something about the X-linked factors. It appears that there are many regions that are quite close to the Xist gene itself that encode non-coding RNAs. These particular DNA elements, which I'll show you in the next slide, are all regulating Xist expression, and we know if we do something to manipulate these different elements, we get the incorrect number of X chromosomes being inactivated. And so they must have something to do with counting. So let's look at those factors now, these DNA elements. So what's pictured here is the X inactivation center. It's around about 100 kilobases in size, although this picture isn't perfectly to scale. And at its center is the Xist long non-coding RNA gene, here shown in blue. In the darker blue small box, I'm indicating the repeat A, rep A. And this is the region of Xist which is very important for silencing because it binds to polycomb repressive complex 2, as I've mentioned before, and I will come back to again. But all of these other elements that are shown here, all of these other genes, also contribute to the expression of Xist. They all control Xist expression in some way, shape, or form. So in general, the ones that are shown in the yellow to red tones that are down this end of the X inactivation center are involved in repressing Xist expression, whereas the ones shown here in green, FTX and JPX, are involved in activating Xist expression. Now, what we know is that this X inactivation center, if you take this region in its entirety, is necessary and sufficient for X inactivation to occur. So what I mean by that is if you delete this region, then X inactivation won't occur, therefore it's necessary. However, if you take this X inactivation center, this whole region, and place it onto an autosome, then you will get inactivation of at least part of that autosome. And so it's sufficient to get this inactivation to take place. We know that we need more than just the Xist gene itself. Instead, we need this whole region with all of these other elements. This can also be seen because in some patients there are translocations between the X inactivation center and autosomes. So it's not just through experimental methods that we've placed the X inactivation center on another autosome. So what are all of these different factors doing and how are they controlling Xist expression? Well, in all cases they are long non-coding RNAs. So this center is made up of a bunch of non-coding RNAs which control another non-coding RNA. I'm not going to go through the specialized features of each one of these, but perhaps the most important one to remember is TSIX or T6. This long non-coding RNA is transcribed antisense to Xist, and it represses Xist expression. Whereas DXPAS34 and XSite themselves activate T6 and thereby indirectly repress Xist expression, because they do so by activating T6. So again, these precise details aren't important, except to remember that we have a predominance of factors which repress Xist expression with just a couple that activate Xist expression within this X inactivation center, all of which are long non-coding RNAs. So if we look upstream of this X inactivation center, a couple of 100 kb upstream, we find another gene called RNF12. And this gene RNF12, rather than being a non-coding RNA, actually does code for a protein. The protein is called RNF12 or RLIM. It has two names that are interchangeably used. So this gene RNF12, being that it's on an X chromosome, is subject to X inactivation. So what's very interesting was that in 2009, a real breakthrough came in our understanding of X chromosome counting. And this was a discovery for this gene, for a role for this gene RNF12, which sits a few hundred kilobases outside of the X inactivation center. So what was proposed and shown was that you needed a threshold level of RNF12 for X inactivation to begin to occur. So I'm going to explain this in a series of diagrams. So to start out with, if you consider that you have cells with 2 active X chromosomes, then the female nucleus has 2 copies of RNF12 and the male has 1, and therefore with 2 active X chromosomes you'll have twice the number of molecules of RNF12, here shown as a red blob. So this is RNF12. You'll have twice the number of molecules in a female nucleus that has 2 active X chromosomes compared with a male nucleus which has only 1. However, then once X inactivation proceeds, you will inactivate one of those copies of RNF12 and then have only the same amount of RNF12 as in a male cell. And so this is why it was proposed that there must be a threshold. So if you had what was shown here as 8 molecules of RNF12 protein, that would be above the threshold and X inactivation would be initiated. Whereas once you have 4 copies as shown now, this would be insufficient to initiate X inactivation. However, although this was really a groundbreaking discovery, it still isn't enough to explain the mechanism of X chromosome counting completely. And that's for 2 reasons. First of all, we know if you completely delete RNF12 in a female cell, X inactivation will eventually occur. It will occur much later than it should have done, and it occurs very inefficiently, but it can occur in some instances. And so this suggests that there are indeed other factors that are still important in counting, and that these can in some way compensate for a loss of RNF12. So the question is then, how does RNF12 actually work? So how is it really going to bring about this initiation of X inactivation? So what we think is that RNF12 activates Xist. So just like those other 2 green elements, those long non-coding RNAs, RNF12 also activates Xist expression, but this time as a protein activator of Xist expression. We think that male cells have insufficient RNF12 to activate Xist, and after X inactivation proceeds within a female cell, we believe that we've reduced the level of RNF12 to such a level that the second X chromosome will not be inactivated because we are below the threshold level of RNF12. So clearly, RNF12 will somehow be involved in this counting process, this first stage of X inactivation, but we still don't know, and this is by the levels of its own expression, this threshold, but we really still don't know anything about the autosomal factors involved, and we don't know anything, and it's clear from the RNF12 results that there are other factors that are also involved in counting, but they're yet to be identified. In the next lecture, we'll think about X inactivation and pluripotency, which I'd mentioned in earlier slides, where there are 2 active X chromosomes in the pluripotent cell types.